Okay, today we'll be looking at a seminar presentation on mutation. These are my outlines: causes of mutation, classification of mutation, mutation detection method, diseases caused by. As a way of definition, mutation are changes in DNA that affect genetic information. It is a permanent alteration of the nucleotide sequence of the genomic of an organism. So this is like normal way an organism is. You notice that the DNA, the proteins forms the trait of an organism, which traces back to the DNA and the mRNA for passage informations. So causes of mutation can be from a spontaneous mutation, error-prone translation synthesis, errors introduced during DNA repair, and induced mutation caused by mutagens. These mutagens can be from an environmental source or from teratogens that have been introduced into the environment. For a spontaneous mutation, this is a situation mutation just occurred as a result of, you cannot really pinpoint what caused or led to this cause or led to the cause of this mutation. We have tautomerism, depurination, deamination, slip strand mispairing. For the tautomerism is as a result of repositioning of the hydrogen atom, while for depurination is as a result of the purine base that is being changed to form apurinic sites. Then for the deamination, we have the keto group is being replaced by an amine group in the DNA sequence. Then for the strip strand mispairing, we have the denaturing of new strands from template during replication by etc. Then causes of mutation, we mentioned the error-prone replication. For the error-prone replication, it is as a result of DNA trying to replicate itself. So it's also a spontaneous mutation. Then we have the other ones that can be introduced by mutagens, which I mentioned before. So some of the mutagens can be as a result of radiations like UV and chemicals from hydroxylamines. Okay. um for We have types of mutations. The types of mutation can be as a result of effect on structure, effect on fitness, effect on function, and then effect based on impact of the protein sequence. Effect based on structure can be in a small scale, large scale, or point mutation. Effect based on function can be as a result of loss of function, gain of function, dominant, negative, litter and reversion. While on fitness, we can be for the fitness it can be beneficial or it can be for this place that this is a small, if there's addition of adenine, it's situation. Insertion and add-ons or more extra nucleotides into the DNA, and it can be reversed. But if it's an insertion situation, it can be reversed. But for a deletion situation, it cannot be reversed. So still talking about the types of mutation, this picture is going to help us understand what we mean by point mutation, deletion, insertion, frame shift. These are types of the mutation. This is a normal gene. For the point mutation, there's an introduction. The G here is being removed, and another alphabet is introduced, which is 18, thereby changing the word dog to a dot. It's a point mutation, a single point mutation. Then for deletion, we notice that the dog is totally removed from the sequence, that's from the sentence. Total removal of the dog. Okay. For insertion, a new sequence is being introduced, which is fat. Fat is being introduced to the dog. So is that's what we mean by insertion. For the French sheet, you notice that the whole word is being turned around. Instead of dog you have ogh. Instead of heat you are having ITT. So that's what we mean by French sheet mutation. So these are the pictures of the type. Duplication, you are introducing new. Inversion is being inverted upwards, upside down. Deletion, a sequence is being deleted totally, or a word is being deleted totally. Insertion, and then effect on functions. We say there can be a loss of function, there can be a gain of function. By a loss of function, you are activating a mutation. Then by a gain of function, you are activating a mutation. And then effects on fitness, we have the harmful mutation, beneficial. Some mutations can happen, it can become beneficial, promotes the traits or fitness of an organism. A mutation can happen and it can lead to the decrease in the fitness of an organism. Some of this you take it through changes in history of an individual, or in a change in the environment. That the theory of use and misuse, where we have we can keep using something that, if it becomes beneficial in the environment, it can be transferred to the next generation. But if we are not using that, if we are not using that structure, it becomes a recessive gene in the sense that it disappears from a generation. So we have neutral mutations and nearing neutral mutations. Impact on protein sequence. Mutation has impact on protein sequence, whereby you have friendship mutation is caused by deletion and insertion of a number of nucleotides. Is not evenly divisible by 3 from a DNA sequence. Nonsense mutation is the point mutation in a sequence of DNA that results in a premature stop codon. Missense mutation is where a single nucleotide is changed to cause substitution of a different amino acid. Example, sickle cell and okay. These are some methods that you can use to detect mutations. We have denaturing gradient gel electrophoresis, constant denaturing gel electrophoresis, temporal temperature gradient, and then diseases that can be caused by mutation. We have the cystic fibrosis, cancer, sickle cell anemia, tay sachs disease, and blindness. In conclusion, mutation that causes changes in protein sequence can be harmful to an organism. On occasion, the effect may be positive in a given environment. In most cases, disease from mutation are incurable and can lead to death. As such, researchers trend towards how to detect, cure, cure diseases related to mutation references. Thank you for listening.